Now, Moderna went public last year, and it turned out to be really the biggest biotech IPO ever. Now, I caught up with the company's CEO, Stephane Bancel, and we talked about what's changed since that IPO.
I think the big difference for us and for our employees is we used to share data all the time, in real time, with everybody. Because to do great medicine, you need to do science, and to do science, you need to have people understanding all the pieces. So that's the biggest change.
So you're sharing more data?
We're sharing less data.
Or sharing less data?
Yeah, because now we have to be careful of when we share data.
Because you're a publicly held company.
Correct.
That's interesting. What's changed, though, in terms of the work that you're doing and the perception with some of your partners? Has anything along that line changed?
So I think this is mostly the same. We used to have partners for many, many years. Our first partnership with AstraZeneca goes back to 2013 and Merck 2015. And we've had a lot of long-term investors like Fidelity and those type of very long-term investors for a long time. So it's the same for us.
Well, I guess what I wonder is, there is something different though, right? Between being privately held and then you become a public company, right? You do have to answer to investors. And I think there's been a lot of talk about your valuation. Bam, out of the gate, it was over $7 billion. I'm just curious, what kinds of questions are you getting more pressure from investors who are saying, they understand the biotech world, right? Things take a while, but you are public now.
I think, again, because we have had for many years very high quality long-term investors, the story has been the same, which is we believe we have a technology that is to build a new class of medicines. We can do medicines that cannot be done using old technology like small molecule, traditional pharma, or biotech products. And so what is very unique about messenger RNA is that it's an information molecule. Every time we actually inject in a human, and we have done it more than 1,000 times for 1,000 different healthy subjects or patients that we dosed already. Every time we inject a human, we basically give them an instruction for their own cells to make the medicine.
Right.
And because of that, we have a platform. And because the human genome has been sequenced now, so we know all the instructions for all the protein we have in our bodies. And so we can take that information into basically our technology and very quickly give an instruction to your body of making protein you need to be healthy again.
Well, you say very quickly. How quickly, though, is it proceeding, right? Because any kind of pharmaceutical, if you will, as you know better than most, it takes several procedures in different stages in terms of clinical testing. And I think about the valuation, and at some point investors are going to say, are we in stage 3? Where are we in the process?
Yeah, so on the speed side, you want to think about two things. One is research, what we do in the labs to understand the drug before it's ready to go into clinical trial. So in the labs, we are able to go very, very fast because the human genome has been sequenced. And because our technology is always the same, we don't have every time to invent how to make the product. It's the same every time. The only difference is the order of the letters on our message, like you have zeros and ones on a piece of software.
So same process in terms for going after the protein.
Correct.
Or creating,
Yes, so if you look today, we have 20 different drugs in development, many more in research, but all of those are made using the similar manufacturing process. And so we don't have, like I used to be in big pharmaceutical companies where for every drug you have to invent how to do it. And so you can go very, very fast.
Can we make, though, the assumption that the efficacy, though, is going to be the same in every process, even if the process of in terms of how you deal with an individual's body, but can we assume the efficacy is going to be the same?
So it's tough to say ahead of seeing the data because we're doing science. But the piece that is interesting is that because you make your own protein, and it's a protein that is known to your body.
Right.
Because it has been in humans.
It's like a custom medication.
Correct, and it has been in humans for millions of years. And so we're not inventing a new molecule. If you think about traditional pharmaceutical, wonderful drugs like Lipitor, Prozac, those have been invented. When we inject an instruction in your body, is to make a protein that you're already making, that is natural in humans.
But and this is not going to come as a new argument to you, because many people have talked to you and challenged you. I mean, it's kind of the holy grail, right? This whole idea of using the body to create a treatment, a pharmaceutical, if you will, or not really a pharmaceutical, a treatment to deal with some disease or ailment. But people have been working on this for such a long time. What makes you think you have the magic sauce to make it work this time?
So I think we are resting on the shoulders of giants. A lot of the technology we are using at the company has been invented by others over the last 20, 30, 40 years. Just one example, we use next-gen sequencing. So we use machines to be able to sequence the human genome. This we did not invent, but we are using those machines that are giving us information that we can then use to go into the technology. And so there's a lot of technology that come together to make Moderna possible. I like to say that 10 years ago or 15 years ago, Moderna could not have existed.